All right, hello. So welcome everyone. So in this video, in this lecture, what we're going to look at is we're going to look at different types of tissues. So tissues are made up of multiple different types of cells. Okay, so you have to have at least two different cell types that are working together. And as these two different cell types are working together, they're working together for a common function. All right. And so what we'll be using today is we'll be using this website called Histology Guide. And Histology Guide is a really good resource for studying and viewing various images of histological tissues. You can break it down by type of tissue, so epithelial, connective, nervous, muscle, or you can break it down by system, so the endocrine system or the respiratory system, and then look at the specific tissues that you could expect to find within that particular system. So what we're going to do in this video is go over just briefly how we can define different types of tissues, and we'll look specifically at epithelial tissues, and then in subsequent videos, we'll go into more detail on different types of tissues. So if you use Histology Guide, you'll navigate the slide box, and you can go here to epithelium, right? So this is one of our four main types of tissues. So we have epithelial tissues, we have connective tissues, we have muscle, and we have nervous tissue, okay? So if we look first at epithelial tissues are going to have a number of different features, okay? So if we click here, we can classify epithelial tissues based on two things, okay? Primarily two things. Their shape, so the shape of the cells, and then the number of layers of cells. So the first thing that you can look at is the shape of the cell. Second thing you can look at are the number of layers of the cells, all right? When you look at the shape, these shapes are either going to be flat, they're going to be cube or kind of box shaped, or they're going to be rectangular or columnar shaped, right? So we have squamous, we have cuboidal, and we have columnar, and that's what you can see here in these rows. So here we have examples of squamous epithelial tissues. Here we have examples of cuboidal epithelial tissues, and then of this bottom row, we have examples of columnar epithelial tissues. You can also classify it based on the number of layers. So you can have one layer, in which we're going to call it simple. You can have two layers or more, so at least two, so two or more, in which case we'll call it stratified. Now, we also have some kind of not exceptions, but kind of some unique cases over here. So stratified is this column here, right? So this column here, multiple layers. This column here is simple, one layer. We also have one called pseudo-stratified. So if you look at the term pseudo means fake or false, right? Like, so pseudoscience is fake science. Pseudo-stratified means that this is falsely stratified. And what this means is that it gives the appearance of being stratified. It gives the appearance of having multiple layers, but in actuality, it's really only one layer. And so to be truly simple, every cell has to touch the basement membrane. And so if you follow all of these blue cells here in this pseudo-stratified example, if you follow them all down, they're all attaching to the basement membrane down here, which is this purple layer way down here. So this is actually simple, but we call it pseudo-stratified instead of true simple. So this would be simple columnar. This would be pseudo-stratified columnar. When we get into tissues later and we look at specific examples, we'll see one way of how to differentiate between simple columnar and pseudo-stratified columnar. And then the other example here is transitional. So transitional, you have multiple layers. They start off kind of round or cube shaped, and then as you get up and as you go toward the apical surface, they become progressively kind of larger and more oblong. Okay, and this is what we'll find in parts of our urinary tract. Okay, so just as a recap, we have shapes of cells: squamous, so flat, cuboidal, cube-shaped, and columnar, column-shaped. And then we also have layers. So we can have simple, we can have stratified, and we can have pseudo-stratified, and then transitional. Okay. So while on Histology Guide, if you go to this right panel, you can see here and you can look at some different information. So you can look at the shapes of cells. You can also look at the number of layers of cells as well, okay? So if we go back and we look at some examples, okay? So say we just want to look at a simple epithelia. You can come over here, click on a blue link, and it will take you into a simple epithelium in this case. So here we have a section of the kidney, okay? So this is the glomerulus here, this structure, this round structure is the glomerulus. All around it, we have single layers of cells that completely surround this glomerular bundle of blood vessels. Okay, so this is a simple epithelium. If we go back and maybe we look at the, let's see, let's go to this example. Let me actually go back. Let's look at a simple cuboidal, right? So say we're wanting to look at a simple cuboidal. Well, we know this is cuboidal because when we look at these cells, they're roughly cube-shaped, right? They're roughly kind of boxy, okay? So when you look at this, you'll see this is simple cuboidal. Now, one key thing to look at when you're studying any type of tissue, epithelial tissue, and really any tissue at all that we'll talk about throughout these lectures, is to look at lots of different images of the same type of tissue. So in other words, if you're looking at simple cuboidal epithelial tissues, look at 3 or 4 or 5 different images of simple cuboidal. This way, you're able to key in on and notice key characteristics of this particular type of tissue. When you look at schematics like these here, these drawings, these are very nice because they illustrate the basic appearance that you could expect to find, but you're not always going to have it perfect like this. So not every cell in a simple squamous is going to be perfectly flat. Not every cell in a cuboidal epithelium is going to be perfectly cube-shaped, okay? So studying these schematics and these drawings here, these diagrams, are good to start off, but then as you start to study and learn specific tissues, you should look at multiple different images of the same tissue type, right? So if we come down and say we look at a simple columnar, okay, and we go to parts of the digestive tract, okay? So if we zoom in and we look here, we'll see that these are roughly column-shaped cells. But if you go around enough, you'll probably find some that look somewhat cuboidal in shape, kind of roundish and cube-like. But this is an example of a simple columnar epithelial tissue, okay? So this lecture, let me tell you one more thing about basic epithelial tissues. They also have a polarity to them, okay? So these cells are going to have a polarity, meaning that they're going to have a top and a bottom. They're going to have a directionality to them. So just like we have poles of the Earth, right? We have a north pole and we have a south pole. We have poles of a magnet. We have a positive and a negative pole. So too, we have poles of epithelial tissues. So this means that we have a top region of a tissue and we have a bottom region of a tissue. The top region that's closest to the external environment is going to be called the apical surface, okay? So all epithelial tissues line the external environment, or they line an opening to a tube or an opening to a passageway. So if you touch your skin, the outside of your skin, this is an epithelial tissue. If you run your tongue on the inside of your mouth along your cheek, that is an epithelial tissue. The food and drink that you swallow that goes down your esophagus, as it goes down your esophagus, it's in contact with the epithelial lining of your esophagus. The inside of your stomach, that's an epithelial tissue, okay? So that which is exposed to the environment, exposed to the opening, the lumen of that tube or passageway, that is called the apical surface. The surface or the region that is in a contact with the bottom, with the basement membrane, that is called the basal surface. And then the lateral side, this is called the lateral surface. So you have the basal part and the lateral part. Sometimes these are grouped together and referred to as the basolateral surfaces, right? So you have a basolateral surface here on the sides and the bottom, and then on the top you have an apical surface, okay? Now on the apical surface, you can have what are called apical specializations. And so these apical specializations are going to be things that you can expect to find on the surface. So here, if you look at this example, this looks like this cell may have, or this epithelial tissue may have cilia, okay? They may also have microvilli as well, okay? So cilia, when we get into later epithelial tissues, cilia help to move and propel things over the surface of the cell, while microvilli serve to increase the surface area that is available for absorption. So you find this in things like the digestive tract and in places like the renal tubules as well, when we're trying to absorb and reabsorb different things, okay? So in future lectures and in future parts, we will look at specific examples of epithelial tissues, and we'll come down here to simple squamous, cuboidal, columnar. We'll look at pseudostratified columnar, and then we'll get into stratified epithelial tissues as well.